Lecture Unit 3, Cells, Part 5. So just a couple of explanations. So the big loud crash you heard in the previous audio was our laundry line falling down. We use a shower curtain rod as our laundry line inside the house, and it fell down. And then the other thing that I heard in listening to the first part of the audio is kind of this muffled background noise, and I want to apologize. That's our cats. When I'm doing these recordings, they love to jump up with me and are trying to get me to pet them, and I didn't realize that they had made so much noise. So I'll try to stop that from future recordings. Okay, folks. So last time we had finished with a description. We'd gone through cell membrane, cell wall, glycocalyx. We had talked about fimbriae, flagella, and the sexpillus. And so now we're going to go inside the bacterial cell. So the inside of the bacterial cell, I refer to this as a cytoplasm, and it's rich in water, and then we have other structures that we'll be talking about: the chromosome, the ribosomes, plasmids, possibly endospores. There's going to be nutrient molecules and ions and waste molecules in the inside of the cell. We want to remember that most bacteria have a single circular chromosome made out of double-stranded DNA. And again, folks, because we'll probably see this cartoon on the lecture exam, this little tangle looks like spaghetti. This is the single circular chromosome. And then this is going to be our plasmid, extrachromosomal DNA that carries extra genetic information such as antibiotic resistance genes. The little dots are the 70S ribosomes. And then this oval structure over here is luckily a relatively unique structure made by just a few groups of bacteria, the endospore, just to give us the overview. Okay, so we have the single circular chromosome made of double-stranded DNA. It encodes the information for all the proteins that the little cell will make, and then many of those proteins are going to be enzymes, and the enzymes will make the nucleic acids, the lipids, and the carbohydrates. In addition, some bacteria will have what we call extra chromosomal. Extra means outside of the chromosome. Extra chromosomal DNA, and they're like little mini circular chromosomes, and these are the plasmids. Now, some bacteria have no plasmids. Other bacteria might have one or more plasmids, and generally speaking, they carry extra genetic information that might help the bacterium survive in a particular environment. And the plasmids we're worried about are the antibiotic resistance plasmids, or R plasmids. These plasmids can carry genes for resistance against 1, 2, 3, 4, 5, or 6 different antibiotics. And then the bacteria can make copies and pass copies of the resistance plasmids to their neighbors. And this is how antibiotic resistance can spread rapidly through a population of organisms, especially in an environment where lots of antibiotics are being used, for example, in hospitals. Now, this is really important, the prokaryotic 70S ribosome. Okay, so all prokaryotes have 70S ribosomes. In contrast, eukaryotes, such as ourselves, we have 80S ribosomes in our cytoplasm. And the reason this is significant is this means the prokaryotic bacterial ribosome is a unique target. We can find drugs that will bind specifically to the bacterial 70S ribosome and stop protein synthesis. But those, if they're good magic bullets, they're gonna inhibit function of the bacterial 70S ribosome without shutting down protein synthesis by our larger 80S ribosomes. Okay, so the ribosomes, they're the sites of protein synthesis or translation. They're made up of ribosomal RNA and proteins. And you might recall, folks, when we were talking about protein synthesis, it's a ribosomal RNA that's the organic catalyst that helps with the peptide bond formation. So we call catalytic RNA ribozymes. The ribosomes are made of 2 subunits, and I know this addition here, folks, it looks crazy, but there's not a mistake here. So in the bacterial 70S ribosome, there's a small subunit that's called 30S. There's a large subunit called 50S, and I know this looks like the math is wrong, but this is actually correct. So the S stands for Svedberg unit. This is the unit we use to measure sedimentation rates when we spin things in really high speeds in an ultracentrifuge. So for lecture exam 1, folks, just remember that prokaryotes, bacteria, have 70S ribosomes, and eukaryotes, such as ourselves, have 80S cytoplasmic ribosomes. And again, the 70S ribosome is a great target for us, right? If we can find antibiotics that bind to the bacterial ribosomes and stop protein synthesis, at the very least, that will stop the bacteria from growing. Another huge advantage is if bacteria are making protein toxins, exotoxins, the ribosome inhibitors will stop toxin production. And in some cases, these antibiotics can actually kill the bacteria. So for lecture exam 1, folks, if you could remember these groups of antibiotics that target the bacterial 70S ribosomes, so the tetracyclines, the macrolides, and macrolides include erythromycin and azithromycin, chloramphenicol, and aminoglycosides. Aminoglycosides include gentamicin, kanamycin, just as a couple of examples. Do note that usually chloramphenicol won't be used to treat a bacterial infection unless it's truly life-threatening. And the reason is in some people, chloramphenicol can cause a shutdown of function of the bone marrow, so you end up with an aplastic anemia. So the purpose of this table is not to make you stressed out, but again, just so we can compare the prokaryotic 70S ribosome to the eukaryotic 80S cytoplasmic ribosome. And again, the antibiotics we use to inhibit the 70S ribosome should not, at safe therapeutic levels, it shouldn't shut down protein synthesis on 80S. Don't worry about the subunit sizes for lecture exam 1. So I think this is the last internal structure we'll be discussing. It's the bacterial endospore. And luckily, folks, there's just a few groups of bacteria that have evolved the ability to make these endospores. And the two groups of bacterial pathogens that can make endospores are those that belong to the genus Bacillus. And the most famous one is Bacillus anthracis, which causes anthrax. And then there's several species of the genus Clostridium that will also make endospores. So for example, Clostridium tetani that causes tetanus, Clostridium botulinum that causes botulism, Clostridium difficile that causes diarrhea and a pseudomembranous colitis. So what are the endospores? They're oval, tough, resistant resting structures. They form in a process called sporulation within the metabolically active bacterium, and in some cases they'll cause like a swelling here. The location of the endospore can be helpful sometimes in identifying the bacteria. So this is Clostridium tetani, and this swelling here at the terminal end of the bacterium, that's the endospore. So the process of forming the endospore is called sporulation. And it's usually triggered when environmental conditions are getting bad, meaning that this metabolically active bacterium is probably going to die pretty soon. So the bacterium will make a copy of its chromosomal DNA, and in the process of forming the endospore, layers of peptidoglycan and proteins are laid down in layers around the chromosome. The endospore has very, very low water concentration. It has a high concentration of a substance called dipicolinic acid, and it has a high calcium ion concentration. And for reasons that I'm not sure we totally understand, the structure, the composition of the endospore permits the endospore to remain infectious in the environment for decades, centuries, some people say thousands of years. So what will happen eventually is when the environmental conditions get back, let's say all the water dries up, there's no food, the metabolically active bacterium will die, it'll break open lice, and it will release the endospore into the environment. And again, those endospores can remain infectious for many, many years. When environmental conditions get better, then just like a little seed, the endospore will undergo a process called germination, in which the endospore, it breaks open and releases a brand new metabolically active bacterium. Now, the endospores are bad news because they're often the infectious forms for infection. So it's anthrax endospores that we inhale or we swallow or get into a cut that will then lead to the development of inhalation anthrax. gastrointestinal anthrax or cutaneous anthrax. Endospores that get into anaerobic areas. For example, the Clostridium tetani endospores, if they contaminate the umbilical cord, the cut umbilical cord of a newborn baby, the umbilical cord, after it's been cut, there's no more blood supply, so it becomes anaerobic. And we'll find out the Clostridium bacteria can only grow anaerobically without oxygen. So if our tetanus endospores contaminate the cut end of the umbilical cord, the endospores germinate, and then the metabolically active bacteria start pumping out tetanus toxin, and that leads to the to tetanus. This is when all of our skeletal muscles contract at the same time, and that can paralyze the muscles used in breathing, and we can die from suffocation. This is a huge concern in newborn babies, right? So if, for example, the instrument used to cut the umbilical cord literally is dirty, because this is where endospores hang out, is in the dirt, then the poor little baby can develop neonatal tetanus. Now, I think this baby was obviously at a hospital with respiratory support, and so we're keeping our fingers crossed that that baby survived. Of note is if mom was current on her tetanus vaccinations, if she'd gotten her booster every 10 years, mom would have passed protective maternal antibodies across the placenta to the baby. And the antibodies against the tetanus toxin, called antitoxin, as the endospores germinated in the umbilical cord and the bacteria pumped out the tetanus toxin, the mom's antibodies circulating in the baby would have bound to the tetanus toxin and prevented the tetanus toxin from binding to the baby's neurons, and that would have prevented the baby from developing tetanus. So again, you guys, I'm a big supporter of vaccination, and really important that moms that are pregnant are up to date on their vaccines so they can pass those protective antibodies to their babies. This is just a cartoon of the steps of endospore formation. So here's a metabolically active bacterium, copies its DNA, lays down layers of protein and peptidoglycan. So here's our endospores formed here. Here the parent bacterium is dying and breaking open, releasing the endospore here. And then when it germinates, it will release this metabolically active bacterium. So the process of forming the endospore is called sporulation, and the process whereby the endospore releases the new metabolically active bacterium is called germination. Just like I think of a little seed, a little plant seed germinating. And this is just a better cartoon of the, here's our, oh, sorry, oops, doggone it, let's go back here, okay. This is our, this is just a better cartoon showing germination. Here's our endospore, and here we see the process of germination where the metabolically active bacterium is released. The endospores, oh my gosh, this is so important. So endospores are really, really tough and resistant. They're hard to gram stain. So some of you, when we're working with members of the genus Bacillus, you'll see these oval structures inside your bacteria that aren't gram staining. And there's a really good chance those are endospores. The endospores are totally resistant to antibiotics. They're resistant to a lot of antiseptics and disinfectants. As we said, they permit the bacteria to survive in the environment for long periods of time, resistant to drying. They won't be killed by normal boiling, but they will be destroyed by autoclaving. That's why the autoclave was developed, to destroy endospores. And furthermore, our phagocytic cells can't destroy endospores. So if I, for example, had inhaled anthrax endospores and my the macrophages in my lungs, the alveolar macrophages, ingested the endospore, they wouldn't be able to destroy them. So just quiz slides. I'm going to pass on this, you guys. So folks, there's a good chance that I'll be assigning the eukaryotic cell worksheet as homework. Again, most of you have had 2 semesters of A&P, or you will have 2 semesters of A&P where you're going to study humans, which are eukaryotes. So I might assign the eukaryotic worksheet as homework, and I'll try to let you know on Canvas before our lecture exam which topics you're most likely to see on eukaryotic cells, so you'll have a little bit of a heads up. So I'm going to let you work through these slides, these last slides. So in general, eukaryotic cells in general are larger than prokaryotic cells. And just really quickly, you guys, we'll just run through some of the eukaryotic microbes we'll be looking at in lab. So we'll be looking at some animal-like protists, which we know are eukaryotes. We'll be looking at Plasmodium. Maybe as a demo, we'll look at Balantidium. This causes diarrhea. Plasmodium is the cause of malaria. This is just a little bit of detail on how we think the eukaryotic cells evolved from prokaryotic cells. Infolding of the cytoplasmic membrane eventually evolved into the nuclear membrane, and the endomembrane system, for example, the endoplasmic reticulum and Golgi body. This is important to know, folks, the endosymbiotic theory. So you want to know the endosymbiotic theory states that eukaryotic mitochondria and chloroplasts evolved from primitive endosymbiotic bacteria. That's important to know. And this is some of the evidence that supports endosymbiotic theory. So eukaryotic mitochondria and chloroplasts, this is really kind of cool. They have the correct size, the same size as bacteria. Very cool. Mitochondria and chloroplasts have 70S-like ribosomes, just like bacteria. Mitochondria and chloroplasts have circular self-replicating DNA, as if this was like a little primitive bacterial chromosome. And the membrane arrangements of mitochondria and chloroplasts support the hypothesis that at one time these were bacteria that lived inside of a primitive nucleated cell. This is the process of phagocytosis. This would have been the process by which those primitive those primitive bacteria would have been brought inside a primitive nucleated cell, and the only difference being, instead of being destroyed, those bacteria took up housekeeping inside the nucleated cell and become endosymbionts, so a symbiotic partner living inside, and they replicated and were passed down when the nucleated cell divided. This is just showing an amoeba that looks like it's phagocytizing maybe algae or cyanobacteria, so that might have been similar to one of the events that led to the evolution of eukaryotic chloroplasts from primitive cyanobacteria. We will talk in lab, folks, that structures used by eukaryotes for movement include flagella. So we'll be looking at the beautiful euglena with this beautiful flagellum that whips back and forth. Doesn't rotate like a boat propeller, but whips back and forth. The eukaryotic flagella and cilia. The cilia will make note of those on another really cool protozoan, paramecium. Although they're used for motility, just like bacterial flagella are, the structure is really different. Those of you in A and P probably remember if we did a cross-section through the eukaryotic flagellum or the cilia, they have what's described as a 9+2 arrangement of microtubules, and then the flagellum is covered in an extension of cytoplasmic or cell membrane. The bacterial flagella is just naked protein. This topic, you guys, I'm going to skip because I'm pretty sure we're going to run out of time to discuss it. We will come back to this topic, talking about ciliated cells of humans in the upper respiratory tract, the trachea, and in the oviducts or fallopian tubes. And we'll talk about how these ciliated cells, they beat to create currents to draw substances across the surface of the cell. And then we'll talk about how microbes can cause damage to these ciliated cells and what kind of serious consequences that can have. But I think we'll delay this and we'll talk about it in our immunity section later on in the course. Another structure used for motility by eukaryotic cells are flagella, oh my goodness, pseudopods, false feet. And we'll see these in our amoeba in the lab. The pseudopods are used not only for motility, but they're also used in food acquisition. So the pseudopods will attach to a food particle, and then the pseudopods can fuse around the food particle, bringing the food particle into the cell in what's called a phagosome. And we'll see that our phagocytic cells, our neutrophils and macrophages, use the same process to ingest and try to destroy invading pathogens. So the plasma membrane, folks, this will also be part of the worksheet, but I can do this really quickly. This is going to be so simple compared to what you all learned in A&P. So we want to look at how cells move substances across their cell membranes, because this will be really important. So first of all, you guys, if we talked about two major types of transport across membranes, there is passive transport, and this is when the substance is being moved from an area of higher concentration across the membrane to an area of lower concentration. It's passive, it doesn't require any extra energy. It may or may not require a membrane protein. And then in contrast, folks, the next slide, I believe, will show us active transport. And this is when the cell is moving a substance against its concentration gradient, meaning the cell, excuse me, is moving the substance from an area of lower concentration across the cell membrane to an area of higher concentration. This always requires extra energy. For example, it might involve spending, quote unquote, using ATP, and it always requires a membrane protein. So let's just back up and look at this slide. I like this slide because it shows all these different types of passive transport. So again, folks, we're moving from an area of high concentration to low, high to low, high to low, high to low. And consequently, just the kinetic energy of the molecules of substance is sufficient. That's going to supply the energy for this transport process. So no additional energy is required. And we can see in this cartoon, folks, some substances can be passively transported right across the phospholipid bilayer. So diffusion, non-polar molecules like molecular oxygen, CO2 could just diffuse right across the phospholipid bilayer. But in contrast, if we have large polar ionized hydrophilic substances, they will require a membrane transport protein to help them. And in this case, the transport is called facilitated diffusion. Facilitated means to help, so we have a membrane protein helping. This is real simple. Looks just like a little water-filled channel here. And then our large hydrophilic molecules, if they have the right size, the right charge distribution, the right shape, they can pass through this water-filled channel from one side to the other. So facilitated diffusion. This is another example of facilitated diffusion, a little bit fancier in that the molecule that's going to be transported has to fit, has to have a complementary fit to kind of this binding site right here. Once it binds, the protein changes shape and opens up and releases the molecule to the other side. But again, it still involves a membrane protein and it is passive, so facilitated diffusion. I was so glad when they finally discovered and started talking about aquaporins. So again, these are membrane proteins and has a channel so that water can move down its concentration gradient from high to low. In the old days, they would say water would just diffuse through the phospholipid bilayer, and to me that made me crazy. It's like, what would attract the water molecules into this hydrophobic area? So it made me really glad when they started talking about aquaporins involved in osmosis, the movement of water from an area of high concentration to low concentration across a semi-permeable membrane. This last slide, folks, is on active transport, and those of you that have had A&P, do not worry. I don't ask you about uniport or antiport or symport. I just want folks to know what we mean by active transport. So active transport is when we're moving the substance from an area of low to high concentration, so low to high. Let's see here. This is we're transporting this from, oops, my bad, you guys. We're transporting this molecule from an area of low to high. Right? And this one's going from low to high. And again, active transport, folks, we're going from an area of low concentration to an area of high. It always requires an input of energy. So you can see here the cell is spending ATP in these particular examples, right? And it always requires a membrane protein. Okay. Group translocation, folks, this is only found in prokaryotes, and this is when the molecule that's being transported across the membrane is going to be chemically modified. So here we see in many bacteria when they're transporting glucose into the cell, they'll phosphorylate it. So that's an example of what we call group translocation. We don't see it in eukaryotes. And that's it. So thanks for your patience, you guys. I know this is such a long PowerPoint, but I hope for some of you, at least maybe these little audio recordings are helping a little bit.